the organization committee asked me to give a keynote speech, and they said you have 1 hour to talk. 1 hour to talk is too much, and I haven't had much time. I prepared bunch of slides. It could go more than 1 hour. If I'm shooting close to that, I'll be running like a horse. So forgive me. I have some good fish slide. Fish is not in the pond or river. It's the chromosome, the gene location directly on the chromosome. I hope you will enjoy those. Let me talk about food. We are about 7 billion people at this moment. In about 30 years from now, the population would be more than 10 billion. And the crop production, what we need, somewhere between 70 and 100% increase of present day food to tackle the world. So it's a lot of work to do, man. And we already witnessed, let's say, 2008 or 2009, there was a food shortage around the world, some places, not everywhere. And that led to riots in some part of the world. We already witnessed this. And what would happen in 30 years from now if you are not going to act right away? The other problem is the plant breeding. This is peak for developing high-yielding varieties. Then it will be coming to these people. They will be probably saving the world. It's time to act now. There's no time to sit down and relax. And what we could do, if the plant breeding is there, it's picked. There's a wild relatives, all land races. We could explore those. And then GMO. There is, in my mind, there is no alternative to GMO. It has to come. Think about the UG99. That in Africa, in Uganda, there's a stem rust in wheat. If it happens, the loss would be 100%. Then what would be the impact in the world? And this is what is a stem rust. And that is not my talk. This is the background. And my field is cytogenetics and molecular cytogenetics, that how that can help enhance the breeding program and GMOs too. The loss is, if it's 100%, the crop is gone. And it's originated from Uganda and is spreading all over the world in food growing region. I'll be talking in molecular cytogenetics, in plant breeding and genomics, how that technology helps in this two field. I'll set an example about transferring a mobile character from a wild relatives into cultivated background, and a brief history about GMO. Please ignore me if I misled something about the GMO. I just gathered the information from the web. It's just a brief history. Then I'll be talking about the technology, genomic in situ hybridization. I'll explain what it is that uses in white crosses, and then fluorescent in situ hybridization to localize the DNA sequence or gene or marker directly on the chromosome. That could help in genomic research, GMOs research, and will be how fish works in genomics. Okay, the first example about endosperm novel protein gene. So we know land races or wild relatives as a vast reservoir of useful character. Could be disease resistant, insect resistant, stress tolerant, and then quality food, the protein and bread making quality. The work I did in my graduate research, that was to transfer the high molecular weight glutamine subunit from a wild relative, that is Agilops umbellata, and is this one. And Holdfast at that time, mid-80s, was the one of the best bread making wheat in the United Kingdom that didn't have that very high molecular weight glutenine subunit. The target was to transfer this subunit into this background. We know wheat is allohexaploid, 3 different genomes made that wheat, and that wheat it's evolved naturally. Now the question is, which chromosome should be targeted to transfer that gene? Not randomly any of those 3. Has to be done scientifically, precisely. That it takes time, but that would be the target. 1A that has a null allele, and these 2 allele, that's the gene here that's produce the good bread making quality. So our target should be to transfer the gene from here to here, but that is close to the centromere, and it is hard. And then this slide shows, you see the yield when you substitute all 3 chromosomes for that alien chromosome, the yield got lost. But the bread making quality just for chromosome 1A, it's significantly high. That it shows that should be the target. What is the substitution line? You remove 1 pair of homologs for that alien chromosome, and it is 42 chromosome wheat, and it's removed that 1A from that wheat background. When you cross this one with the pairing regulatory gene, I'm not going to go to details, then this is the situation. We would expect recombination here. When you use the alien species and suppressing the pairing regulatory gene, what would happen? That 3 different genomes, it can mess up. And here it shows it's a quadrivalent. This is pentavalent with the integration segment is here. And this is another quadrivalent, other one, another one. Now we need to clean up. Wheat is a unique crop, second to none. Cytogenetically is highly resourceful organism in plant science. And it has all the nucleotide lines to play with. Now what it shows, the transfer was done, and all the A lines with the glutenine and B line gliding. Gliding was not our target. And it shows that it's got the recombination. Now we need to clean up. So when you do the telocentric analysis, is a 21 bivalents. 20 bivalents are ring bivalent. What ring bivalent tells, there is no residual background here. They pair nicely, and that's why you're getting the ring bivalent. But that the recombinant chromosome, it's a rod bivalent, and it's called ditelotrivalent. So integration is here. So that's what we need. And these are some example of the integrase line. This is the A genome. That's the B genome. B genome was not our target, but it happened that due to that pairing suppressing gene, and also some with 1D. With the sedimentation test for bread making quality, it showed all this A line showed significantly higher than the wheat background. The transfer was successful. Now the question is, sometimes the alien integration, it's large and it takes some undesirable character. We need to reduce that unwanted segment. How we could do that? If you interpose between 2 recombinant lines, you could end up a cryptic translocation or recombination or a small translocation. This is just an outline showing how we could achieve it. Now it's come fish or dish. The principle is you have a DNA, you label that DNA, it could be direct or indirect. If it's direct, then you use the labeled fluorochromes with that DNA segment, or if it's indirect, it could be biotin or dighapten. What happened after this labeling? You have a chromosomal DNA on the slide. You do the same procedure, but little bit different than molecular genetics. You hybridize this target DNA, that's a labeled one, and it will find the target site, specific site in the chromosome. It will not bind anywhere else. It shows here when you use that, and it's just hybridizing towards the end of the chromosome. This is sorghum chromosome. But for this, we use the bacterial artificial chromosome. It's a large chunk of DNA. It's easy to handle. But what the key, you need a good chromosome spread and quality probe. That's what is required. You don't need a brain to do that fish or dish. Fish, it could be used for white crossing in plant breeding, genome structure, organization, evolution. Nuclear organization, gene expression, karyotype development is very important for a new species or organism we work with. We need to know how they look like. What is the complement, chromosome complement number? And what are the centromeres of that chromosome? It's important to know. You know that human genome, that the success came from the cytogenetics, and all the genes they discovered so far is lined up along the length of the chromosome, and where it is exactly, they can say it's there. That's it. But in plant science, we're lacking that side, because preparing plant chromosome is a little bit tougher than animal chromosome. That's why, and there is no money. We need to put some money in research. What we do, we prepare the chromosome spread from the root tip, collect the root tip from the growing seedling. We are not killing the seeds. It's let them grow in a pot. And if you need to go back 2-3 times, say this is an important line, this is a transient line, we don't want to lose that. But we want to find out where the gene is inserted. You can go back to your greenhouse and collect many times you want to. And then you find out what the gene is. This slide shows an example of genomic in situ hybridization. What is genomic in situ hybridization? GISH acronym. You take the DNA from, this is a tritically rye and wheat hybrid. It's man-made 1875. Think about when plant breeding started without science that time. Durum wheat crossed with rye, and durum with 28 chromosome, and rye is 14 chromosome from rye, and they are easily separated in a common background. That was the rye DNA was total rye DNA was labeled, and then detect with that fluorochromes. When you put it under the fluorescent microscope, you can differentiate them. Somewhere along that line, it's got introgressed from the wheat chromosome, but we do not know from which one, but it can be easily identified. A few more examples about the genomic in situ hybridization. That is, this is addition line, this 44 chromosome in wheat background, 6R addition. This is 1B, 1R translocation line. Most of the CMIT, high yielding variety, contain these guys. And it has a profound effect for higher yield. This example of LR leaf frost resistant 24. It's from rye. This is leaf frost 19, and leaf frost 37 is a small translocation. This barred yellow drop virus resistant gene here, and this probably isochromosome. This was done when I was at CIMMYT. Maize tripsicum hybrid. Tripsicum is carrying the apomictic gene. In CIMMYT maize program, French group doing it, their target is to transfer the apomictic gene from tripsicum to maize. This is tryptophan F1. And all the maize 10 chromosomes are separated from the tryptophan 36 chromosome. And the maize chromosome was labeled, and tryptophan DNA was unlabeled. This is an addition line. One day I was doing an experiment running that had a 21 chromosome and did that maize DNA labeled and tryptophan unlabeled. So what it shows, there's a translocation, but we are talking, few people had said it could be an artifact. So if it's an artifact, then I thought, why not go back to the greenhouse, collect some more root, and then use the tryptophan DNA as a labeled DNA and maize DNA as a blocking DNA. If you see the difference, it's tryptophan DNA here, and at the end it's maize DNA. That is the first proven maize tryptophan translocation at CIMMYT. Okay, now let's move on to the GMOs. Its first antibiotic resistant, it's happened in '82, and then so on. Now we are almost lot of crops, 27 crops at this moment. And then first approved GMO for cell 94, then in 95, bunch of them. Interestingly, if you see, most of them are all from the company. And they're profiting from this. Then how would happen if we need the food for the world? They'll be collecting the dollar. But I think the governments all over the world needs to sit down and think about it, how we can work for the future. It's very important. In 2000, there's a golden rice scan, and that was one of the major breakthrough in GMO crops. And then 5 years later, Syngenta produced golden rice 2 that was 23 times beta-carotene than the golden rice 1. And you see the effect of GMO crops. It's profound, and we need it, no matter what. If you see GMO crops cultivated land from 96 to 13, it's about 100-fold. 1.7 million, then 175 million in 2013. Biotech seed cell, global is 15.7 billion, and industrial nation is about 72 and 11.3. And see, who is picking up the food? These fat guys. Is about 7.3 billion from biotech seed, and mostly 50% of their sale coming from Roundup product and GM seed. It's a yield increase. Herbicide tolerance about 9%, insect resistant crop 25%. But this one is offset that increase due to the seed price. It didn't make it, it's just balanced out. But in the developing countries, about 14%. Oh, yes, so many characters have been, traits have been inserted in GMO, and then about 27 crops. In developing countries, that's about GMO crops 54%. Since 94, 2,833 regulatory approvals have been done, and that involving 27 GMO crops and 133 GM events, and then 1,321 are approval for food, direct or processing, 918 for feed. Here is the question to Jen and others, and where is the cytology can play a big role. We all know Bt gene resistance is breaking down, even though the refuses were suggested to do, but the insect is evolving the resistance. So what the fish can do in transgene research, we can catalog them. And then study further to see which one is doing better over the other one and which one is backing down. And that is, in my mind, is important. It's not harming anything. It's just giving one step further in our research. This is an example of tobacco, 48 chromosome. And in most cases, we don't use the cover slip. We take the root tip, do the enzymatic digestion, and take the meristem portion of the root, and then macerate it over this glass slide. And you see all the 48 chromosomes are unifocal. You don't need to focus to see one then another. And even the centromeric position after fish. This is a satellited chromosome, and that's the satellite portion. Transgene is here, but not there, not in the homologous. But there is a transgene location here. This is a DEPI image of the same cell, and that's the satellited chromosome, that's the partner chromosome. This is a Nicotiana bantamaniana. This work with Nobel Foundation. This is code protein gene, about 7 kb. This bantamaniana is, you see, most of them are acrocentric chromosome and few are submetacentric. This one is submetacentric. You know what is submetacentric and what you get acrocentric. A little bit of short arm and mostly the long arm. This is a prophase cell of the same transgene, and it shows it's on the short arm and near the centromeric position. This is from a source. I cannot tell the name of the source. It's a maize transgene, and this is a both chromosome. This is homozygous. This one also homozygous, and this one was not. There's only single insertion. What would happen? It's going to segregate from the next generation. But we need to know where it is, which chromosome, and how they're behaving. It's important. The next phase is how fish can help in genomic research. You know pine. Pine is a giant species, about 22 to 36 gigabases, and it has is fully retrotransposome. This is the Pachytene spread of pine, but I'm going to talk about, just concentrate here. In the Pachytene is two homologous chromosome pair. Here is a loop. What it tells, this chromosome has more chromomeres than the partner chromosome. Here is a gap, here is a gap, here is a gap, here is a gap, and here is a loop. It's gap on this chromosome, gap on this chromosome. Carol Lupstrom, she's a professor at Texas A&M. This is how many, about 98 or 97, 100,000 SNPs. They just found it in that species. And yeah, that is obvious. If you can correlate this one and that one, that makes sense. And you see how science is helping each other. We know pine and chestnut. Chestnut is a big tree, and used to be, it's completely wiped out. I'm going to talk this afternoon about chestnut. It's a giant tree, used to be known as the king of the forest, and taller than pine. And you see how big the pine chromosome, about 25 times larger than chestnut. If we, as I said, karyotype development. If you work with a species, we need to know the chromosome morphology. With techniques improved over the times, this was the first publication in the Dowderic group in slush pine, 95. If you have a spread like this, you cannot develop. This is not a criticize. I'm not criticizing this, but is the technique improved? You cannot make a nice karyotype from there or a ideogram. If you have a spread like this, It's difficult to prepare the point chromosome spread due to their nature, that large chromosome. But it's the same clone we used, and we can easily differentiate all 24 chromosomes. This is loblolly. This is published from New Zealand. You see how stressed the chromosomes are, and this is the same line. And again, it's showing 3 different filters of, this is the telomeric probes. It should be concentrated towards end of the arm of the chromosome, but mostly they're concentrated near the centromere. Evolutionary, it tells me probably the chromosome fused the end, and then the remnant, the telomere is still there in the centromere. You see how strong the signals are near the precentromeric region. And 18S is the red signal from the 18S and the green signal from the telomeric probe. And we use 108 readings for each event to develop that ideogram. So we did the chromosome length and short arm, long arm, then signal distance from here to there, then from this one to end. And we came up with 54 bins, and each chromosome mostly 4 to 5 bins in each chromosome. If we find one, say, marker, and we can analyze this and put it, where is this marker coming from, whether chromosome 1A or chromosome 5, not 1A, chromosome 1 or 5. If it's from 5, where it is located. Is it in the bin or 22, 20, 21, or 25? Here is one example. I was working with, not this one, Dan Peterson, Mississippi State, with repetitive DNA. That one repetitive DNA has a unique signal. It has some retrotransposase as well, but it hybridizes at the end of a homologous pair. And then we went back and we said it could be chromosome 2, 3, or 5. You see the 18S position, and depending on the intensity. Then we did further experiment. We then found out it could be on chromosome 7. So we concluded that repetitive DNA, the unique repetitive DNA of that clone is on point chromosome 7. Fish in chestnut. This is a satellited chromosome. About this portion was not mapped earlier, but we mapped it with this fish experiment using 4 bags. This is first experiment using the bag, and what it shows under the microscope, there is satellited chromosome. Then I thought, why not use the 18S probe and see where it's landing? And when you did that second probing with the same slide, and it showed the 18S probe hybridized there on the satellited nuclear organizer region. And here it's what it is. If you see that 1.3 to about 56 centimorgan unit, and that mass of that chromosome section were not mapped earlier. That is 18S site. And we did all chestnut chromosome assigned each of the linkage group, 12 linkage group or 12 chromosome in chestnut. There's one problem I noticed that, so we should expect this, the genetic map, but some of them are showing this way, upside down. So which one to accept? So we need to sit down and just solve this issue. Fish in sorghum, to see the integration of linkage map in cytogenetics. It's about 247 or 42 unit, map unit. We use 19 probe. All of them 19 probes going towards the end of each chromosome. Here is an example of chromosome 1. You see, it's about 84 map unit or 81, I can't read. Anyway, we took 3 markers from there, and 2 markers go to one arm, and the other mark goes to the opposite arm. And what it tells, these 3 markers spending about 50% of the chromosome. What is that 50% of the chromosome? Is the heterochromatic DNA. You cannot get the recombination there. That's why there is no marker. That tells us it's a big chunk of DNA there. But it's not like this. And this is the actual map for that genome. And it's, I used 28, but 2 didn't work. The back 10 and back 17. But 26 did work. This is the pack-18 chromosome of 3 of sorghum. And we know the gene synteny. All the Gramineae family, they have a gene synteny, say, from sorghum to rice or maize. And it lined up so nicely with the rice chromosome. In the pericentenary, about 40 to 50% of the chromosome, each chromosome do not have the marker, doesn't have the marker. So all the signal, they're going towards the end of the chromosome. In Poplar, did the work with Jerry Tuscan, he is not here today, asked me to find out where is that major 18S site located in that linkage map. First guess was chromosome 15 and 14. Second guess was 4 linkage group, and as you see, then when you did the fish with a number of linkage group back fish, then we identified that chromosome 14 and chromosome 8, there's a 218S site in Poplar that confirm that which linkage group housing that 18S site. The next phase, we added an LCM microdissected microscope. Our plan is to use that microscope is our genomic research. What we could do, whether we can sequence a specific chromosome or chromosome by chromosome, because the idea is, probably all of you know, the genome sequencing is moving so fast that single cell can be sequenced nicely. If single cell can be sequenced nicely, then why not one chromosome, single chromosome? If the single chromosome can be sequenced, then if there is any problem with the assembly, the genome for that specific chromosome, we can say that problem comes from chromosome 1, not for whole genome. And that's the approach we trying to solve that issue. Is brief history about the microdissection. First in Drosophila 81 that used the needle to take the DNA, and then mouse, human in 86. And then the other use would be microdissection, chromosome painting, linkage map, physical map construction, ES degeneration. How to take the one step further in genomics. Y pine, chromosome 12. Chromosome 12 is easily identified. That's the only chromosome in pine is submetacentric, and rest 11 chromosomes are metacentric. It's hard to differentiate. But if you use the fluorescent dye, you can differentiate other chromosomes too. And it's a huge genome, say about 22 gigabases, and then one chromosome about 6%. And we can spread the chromosome so nicely, and then it's easy to use for the LCM work. This is chromosome 12, that's the smallest pair, can easily be identified under the microscope. This is a laser capture microscope. We target the chromosome. The next slide shows, so this is chromosome 12. What happens when you take this chromosome, laser can take up some piece from the nearby chromosome. We need to destroy those with the laser. So like this. So then all the possible contamination could happen. So we just burn that segment and then target that targeted chromosome. Then the laser shoot the pulse and put it in the cap of the microcentrifuge tube. We took the DNA, did the whole genome amplification. It showed it did pick up the DNA. But the issue we found is it has some contamination. Jerry did some work with that laser capture microdissection. We found human DNA there, bacterial DNA there, human DNA, it could be my DNA, because I was taking up the chromosome in that cap. So when it happens that, this is the guy, is my project leader. He was instrumental to buy this toy. It's about $350,000 toy, and it's sitting in our lab. And there's another guy, the head of the Department of Soil and Crop Sciences at Texas A&M. It's USDA, I think, poured about 65% of the money, and university 35%. And he was behind to collecting money from every door. Thanks to them. It's can pick up entire chromosome and rapid and precise, and chromosome-specific region. You can isolate scientific region, short arm, long arm, and then whatever you think that's which portion you can take it out and put it in the slide. The contamination. We designed that table. It's about, I think, 75 inch long and 48 deep. We put it under the laminar flow hood. That is also custom made. So we are making slide in a laminar flow hood, contamination-free area, to minimize the contamination. And then when you will be picking up, the chromosome will be picking up under this environment. So hopefully, it will be less contaminated. Okay, that is almost end of my talk. We do fish, but we also go out fish outside. That's me. You see how many fishes there? It's in the nearby lake. And for food, we grow our own vegetable in the backyard. And we just distribute to the friends and neighbor. And from May to December, we don't go to market to get the food, vegetables. This is, Asian people know what it is. And you see how long, and this is the one vegetable, leafy vegetable. And you see the okra. This is really good variety. If anybody wants seed, I can send the seed of okra. Yeah, that's chili. Well, that's all, and thank you for listening. Any question?
Briefly touched upon the subject of GMOs, sir. I'm from Europe, which is like a very controversial issue.
Oh, yes.
So it's like dividing America and Europe. I'm not here to judge, just a question. In light of modern technologies, apart from whether it really does contribute to the to yield increase, etc.